Continuing AP biology notes on cellular respiration fermentation, we'll now focus on fermentation. So this is anaerobic respiration that allows cells to produce ATP when oxygen is not present. The entire process of cellular respiration requires oxygen to make ATP, but sometimes oxygen is not available for one reason or another depending on the cell. And when oxygen isn't present, then the electron transport chain will not work. So in cells that are capable of doing the entire process, they can undergo a type of fermentation to continue producing a little bit of ATP and keep glycolysis going. And so there are two kinds of fermentation. So in anaerobic respiration, we have an electron transport chain. These are anaerobic respiration that doesn't require oxygen. For example, some organisms might use the sulfate. The fermentation is going to be different than this because rather than using the oxidative phosphorylation or some other type of phosphorylation, the fermentation will use the substrate-level phosphorylation instead of that electron transport chain. There are two main kinds of two common kinds of fermentation, and they are alcohol fermentation and lactic acid fermentation. The main reason for fermentation is to produce reactions that regenerate the NAD+ that can be reused by glycolysis. Because glycolysis does produce some NADH, but you have to have NAD+, the oxidized form, in order to accept those electrons. In alcohol fermentation, we're going to take the pyruvate that's produced from glycolysis and convert it to ethanol. And there are two steps in the process. The first one releases carbon dioxide, and then the second part will produce the ethanol as well as a little more ATP and regenerate the NAD+. The alcohol fermentation that's done by yeast is used in brewing and winemaking, of course, and also in baking. That's what makes bread rise and what makes the bubbles in beer, and the alcohol present in both beer and wine. So in alcohol fermentation, here's what happens. We have glycolysis occurring, of course. We're producing a little bit of ATP here and producing the pyruvate molecule. In alcohol fermentation, again, that like we saw in the oxidation of the pyruvate to acetyl-CoA, we have the removal of a carbon dioxide atom, leaving a 2-carbon chain. That 2-carbon chain is changed, first of all, to acetaldehyde, and then with the loss of 2 electrons, or the gain of 2 electrons and 2 protons, produces ethanol. This regenerates the NAD+ that is needed to continue glycolysis. So we can use the NADH there to oxidize the acetaldehyde to ethanol, and then the NAD+, which is now in its oxidized form, can be used in glycolysis to become reduced and carry those electrons. In lactic acid fermentation, the pyruvate is going to also produce NADH and form lactate as an end product, or lactic acid. And there's no carbon dioxide produced, and there's no alcohol produced. The lactic acid fermentation that occurs by some fungi and some bacteria produces things like cheese, yogurt, sour cream, and so forth. And human muscle cells also use lactic acid to make ATP when oxygen is scarce, as occurs in your muscles when you overexercise and you end up with sore muscles and cramps. So in lactic acid fermentation, again, glycolysis proceeds as usual, producing some ATP and reducing NAD+ to NADH. And here we have our 3-carbon compound that is pyruvate. The pyruvate will accept the hydrogens from NADH to produce lactate and regenerate the NAD+. And so we still get a little bit of ATP production. We can continue ATP production by glycolysis by regenerating the NAD+ and producing some lactate or lactic acid. Now when you compare those processes, okay, you still get a little bit of ATP. All organisms use glycolysis, and all forms of fermentation use glycolysis with a net ATP production of 2 ATPs. And that's going to generate some chemical energy, or use some chemical energy of food, transfer it into a form the cells can use. In all 3 processes, the NAD+ is the oxidizing agent. There are different final electron acceptors in all 3 processes in cellular respiration, alcohol fermentation, and lactic acid fermentation. In fermentation, the final electron acceptor will be the organic molecule, the pyruvate or the acetaldehyde. And in cellular respiration, it will be the oxygen that produces water. In cellular respiration, we get 32 to 36 or 38 ATPs per glucose molecule. In fermentation, we only get 2 ATPs per glucose molecule. So there's a big difference in the amount of energy that is produced via fermentation, but the fermentation is beneficial in that it allows glycolysis to continue and a little bit of ATP produced rather than none at all. There are organisms called obligate anaerobes. Obligate anaerobes cannot stand the presence of oxygen, and they're going to carry out fermentation or anaerobic respiration and will die in the presence of oxygen. It is poisonous to them. There are organisms such as yeast and other kinds of bacteria that are what we call facultative anaerobes. That means they can survive using either fermentation or cellular respiration. So if oxygen is present, they can undergo the whole process and have plenty of energy. If oxygen is absent, they can keep glycolysis going by means of the fermentation process. The pyruvate is the fork in the road that leads to one of two different routes to break down the oxygen, the glucose. Here we see graphically that fork in the road from glycolysis. Okay, if oxygen is not present, then you produce some other product like ethanol or lactate, and if oxygen is present, then the pyruvate can enter the rest of the cellular respiration process and produce more ATP. The one important thing to realize is that glycolysis has been around for a very, very long time. Ancient prokaryotes probably used glycolysis a long time before there was any oxygen in the atmosphere. And it was about almost 3 billion years ago before there was a whole lot of oxygen present in the atmosphere as a result of photosynthetic bacteria. So early prokaryotes probably used only glycolysis. So it's very ancient. Once oxygen was present, a lot of those, frankly, probably died off because oxygen was poisonous to them, except for those few who were able to utilize the oxygen in a different process or do something other than be oxidized and be damaged by it. There are also other ways to produce energy using other nutrients other than glucose. And glycolysis and the citric acid cycle are still major important parts of those processes. There are various kinds of pathways for building molecules and breaking molecules down. The catabolic pathways are going to funnel those electrons in different ways. Okay, and the glycolysis is going to work with a number of different carbohydrates, like not just glucose but also fructose and many other monosaccharides. The proteins have to be broken down, of course, to amino acids, but then the amino groups can also feed into glycolysis or into the citric acid cycle, sometimes as a reactant, sometimes as one of the intermediate products. Fats are broken down to glycerol, which is also part of glycolysis. Fatty acids can be used to generate things like acetyl-CoA. Fat actually produces more energy, about twice as much as ATP as carbohydrate, but it produces other compounds that are not as useful to the cell, and so the carbohydrate metabolism is much more beneficial. Here we see some alternate pathways we have. The amino acids can enter into the chain at the pyruvate level or acetyl-CoA, or even into the citric acid cycle. The sugars, of course, are going to go through glycolysis, the whole process. Fats can enter the process here as part of glyceraldehyde 3-phosphate, which is an intermediate step in one of the glycolysis processes. Fatty acids can be changed or can be converted to acetyl-CoA. So there's a lot of ways that these other nutrients can be used to produce energy and enter the citric acid cycle and eventually produce some ATP. There are also anabolic pathways or biosynthetic pathways that can result from some of these. So some of these molecules, the smaller molecules, can come from food itself through the digestion of food or as products from glycolysis or the citric acid cycle. And all of this stuff is regulated by feedback mechanisms. Okay, feedback inhibition is a very important mechanism for controlling all kinds of processes. If there's plenty of ATP or there's not enough ATP, then the respiration cycle is going to speed up, and when there's plenty of it, it's going to slow down. And the control of this is going to be depend on regulating the enzymes at different points in the pathway. As an overview, let's look at what happens for the whole cellular respiration process just diagrammatically so we can see it. And glycolysis, of course, occurs in the cytoplasm. In that process, glucose splits into 2 pyruvates. It uses 2 ATPs to produce 4 and 2 NADHs, so we get a net gain of 2 ATPs there. If oxygen is not present, then yeast cells and some other bacteria can undergo alcohol fermentation to produce the NAD plus that can be used back up here in glycolysis. In lactic acid fermentation, such as happens in your muscle cells, produces lactic acid and some NAD plus that can go back up here to be used in glycolysis. If oxygen is present, of course, then we'll undergo aerobic respiration in the mitochondrial matrix. The pyruvate is going to be oxidized to 2 acetyl-CoAs, 1 for each pyruvate, of course, and then that's going to enter the citric acid cycle through a series of reactions to produce carbon dioxide, some electron carriers, some ATP, and start the cycle over again. We gain 2 more ATPs this way for each molecule of glucose. We gain 2 NADHs from pyruvate oxidation and 6 from the Krebs cycle for a total of 8 NADHs and 2 FADH2s for each molecule of glucose. All of those energy molecules, the NADH and the FADH2, will travel to the electron transport chain in the inner mitochondrial membrane to power the pumping of protons into the intermembrane space while those electrons are being gradually reduced to a lower energy state. Eventually, the protons will pass through ATP synthase to produce ATP, and the electrons will be passed off to oxygen and water to produce the oxygen to produce water, producing a total of about, again, 30 to 32 to 36 to 38 ATPs total. The final electron acceptor, of course, is the oxygen of water.